Next speaker, of course, is Aurélie Bussy. Aurélie Bussy, for some context, is also a PhD student in neuroscience at McGill University and is working under the supervision of Dr. Mallar Chakravarty.
Yes, so thank you, Olivier, for the introduction. So today I'm going to talk about my project on multivariate analysis of morphometric and quantitative MRI metrics in aging and Alzheimer's disease. Sorry about that. Okay. So as you probably all know, in aging and Alzheimer's disease progression, we can see various neurobiological changes happening. So for example, if we look at the cortical volume, we can see that there is, of course, an increased atrophy with aging. But more specifically, if we look at the cortex, we can see that the regions the most impacted are going to be in the occipital and temporal lobe. And a region from the temporal lobe that is interesting to me, at least, it's the hippocampus. And the hippocampus is also a very key region in the context of Alzheimer's disease, since it is one of the first regions to be impacted by the pathology. And additionally, in this study where they looked at the rate of volume change in mild cognitive impairment participants and Alzheimer's disease participants, so NCI in black and Alzheimer's disease in red in this plot, we can see that the rate of change is faster for the NCI and AD versus control in the gray matter, and that is even more the case in the hippocampus. So then what we also are interested to look at in the brain is the amount of iron. And from postmortem studies, we have shown that there is an increased iron in the lifespan. And we can use qMRI, as we heard this morning, to try to quantify iron in the brain. So here we are looking at R2 star maps of 3 different LC control patients, with the younger in the left and the older participant in the right. And we can see that there is an increased intensity with aging, specifically in the subcortical region, which reflects this accumulation of iron in aging. And if we look at 3 age-matched participants with Alzheimer's disease, we can see that this increased intensity is even stronger. The last thing that I'm interested in the brain is the myelin, and we can see that throughout adulthood, it's pretty stable between 20 and 60 years old. And then we can see that there is a drastic decrease of myelin in older age. And also, there is studies which have demonstrated that there is a specific impact of myelin in Alzheimer's disease. So here they use 4 different metrics which represents somewhat the integrity of the myelin in the brain. And using different metrics, they show consistent structures that are impacted in Alzheimer's disease, such as the occipital lobe, temporal lobe, and the precuneus. So in the context of all of this, there is a few challenges that we've seen from the literature that we want to address in our study. So the first one is that there is very few studies using qMRI to investigate Alzheimer's disease progression. So in order to address that, we're going to use a data set with LC control, people with familial high risk for Alzheimer's disease, NCI and AD patients with standard and quantitative MRI scans. The second thing is that most studies are using region of interest-based segmentation, which are often based on a priori definition for atlases, which might or might not be as relevant for the specific study of interest. So in order to overcome that, we are going to use a data-driven parcellation of the cortex and the hippocampus. And finally, most studies that want to relate qMRI metrics with cognition or demographics are often using univariate analysis. So we are going to try to use multivariate analysis to do so in my study. So in order to do that, we used 158 participants after quality control of all of scans. And here is a representation of the sex and number of participants per group. And here you can see the age range and the median age, which is represented by these vertical lines. So we can see that the median age is similar for the controls, NCI and AD, but we have a younger population for the ones that are at risk for AD. For the MRI protocol, we use the MP2 race sequence in order to extract the T1 weighted scan and the T1 map. And we use a multi-echo gradient echo sequence with 12 echoes to extract the magnitude scans. From the T1 weighted scan, we use the pre-processing pipeline to extract the T1 weighted pre-processed and also a brain mask. We then use CVET to extract cortical information, so the cortical surface, cortical thickness, and surface area at each vertex. we use deformation-based morphometry in order to create this average brain and also extract relative Jacobians that are representative of local variation of volume in the brain. And also we extracted an hippocampal mask. So using the brain mask, we were able to extract our T1 map. And after fitting an exponential decay to our 12 echoes, we obtained our T2 star map. So now that we had all our data ready, we extracted and we created these input matrices where we can see vertex-wise and subject-wise information for T1, T2 star, CT, so cortical thickness, and surface area for the cortex. And for the hippocampus, we had voxel-wise T1, T2 star, and relative Jacobians. So as I mentioned before, we wanted to use a data-driven parcellation in the context of our research. So we used non-negative matrix factorization, or NMF, and that technique was introduced in our lab by Dr. Patel. And this technique, as the name says, has the specificity of having non-negative information in the inputs and the outputs, which makes the interpretation of our results more straightforward, especially in the context of neuroimaging data. And also NMF encourages sparsity, and also it provides spatial components that we can relate to our subjects of interest. So the way it works is that first we need to create our input matrix. So for us, we used our 4 metrics of interest that we concatenated into one large input matrix. And from that input matrix, we extracted 2 output matrices. The first one, which I call a spatial matrix, which gives us vertex-wise components. And the second one gives us a subject matrix with subject-wise metric components. So to give you a better idea of how it looks, I'm going to jump into the results of the NMF for the cortex. So here is the matrix for the subject weight that I'm going to describe just after. And here are the spatial components that we obtained. So we selected 10 components here. So we did a stability analysis to select the best number of components. I'm not going to go in the detail of that for a matter of time. But what you can see from these spatial maps is that we found bilateral components pretty much in all the components. And also they are showing biological relevant regions. So for example, the component 2 is mostly in the frontal lobe, component 5 is mostly in the temporal lobe, 6 will be in the somatomotor cortex, 8 in the precuneus, etc. And the way you need to interpret these results, so for example, if we focus on component 2, which is in the frontal lobe, and in order to relate that back to the matrix, we need to look at the second row. So I just do this row here. And the way it works is that each color represents the amount of weights that each participant has on this component. So darker will mean lower weight and brighter will mean higher weights. And here we will have participant 1, participant 2, 3, etc. for the cortical thickness. And again, here participant 1, 2, 3 for the surface area, etc. So what we can extract is different information. So we can either look at the population base. So we can see that overall we have lower CT and SA in this component and higher T1. But we can also look at more specific inter-individual variations. So if you look at T1, for example, you can see that there is different colors for different participants, and that is what we are interested in to investigate further. So I'm just going to go over quickly. We did the same approach for NMF, this time using GBM, T1, and T2 star. And so this is the weights, subject matrix weights, and we obtained 4 components that are here and shown a little bit more specifically here. So we can see that component 1 is mostly in the body and tail of the hippocampus, component 2 is mostly in the head, component 3 is more lateral, and component 4 is more medial. And now we wanted to relate these components to our demographics of interest. And for that, we used a partial least square analysis, or PLS, which can take 2 input matrices and try to find latent variables that will explain the most variance that it can. So the first latent variables that we obtain explain about 75% of the variance. And the pattern that we found was a pattern of being female, being older, and being in the MCI or Alzheimer's disease group, and having lower airband score, so worse cognition, was related to a brain pattern that I'm going to show. So decrease of CT everywhere in the brain, decrease of SA everywhere as well, decrease of T2 star in these regions in red, and increase of T2 star in the precuneus. And for the hippocampus, we found decrease of relative Jacobians in this component, which is in the body and tail of the hippocampus, increase volume in this more lateral component, and increase T1 and increase T2 star everywhere in the hippocampus. So the findings that were the most significant in our latent variable were the morphometry changes in the cortex, so CT and SA, and the microstructure changes in the hippocampus, so T1 and T2 star. So going into more discussion of the results, what we found was that NMF was able to extract bilateral and also biologically meaningful component. So for example, as sanity check, we could look at this component 6, which is in the somatomotor cortex, and we can see that this region is known to be very heavily myelinated in the brain. And we can see that indeed NMF is able to capture that there is lower T1, which represents higher myelin. So we can see that NMF provides biologically relevant information. And for the hippocampus, we extracted 4 components, which is consistent with the previous work done in my lab. But this time, Rehan was using different metrics, and also the components are slightly dissimilar, but still we found some similarities in these 2 parcellation. Then regarding the results of having higher T2 star in the precuneus, it's actually interesting because we can relate that back to a picture that I've shown during the introduction, where I was saying that the precuneus was one of the regions where they show consistent myelin impairment. And if there is a lot of myelin decrease over time, there will be also a decrease of iron at one point. And therefore, this increase of T2 star is maybe what we can use to capture these changes. And also, if we're looking at the changes that we've seen in the hippocampus, for example, this decrease of volume in the body and tail of the hippocampus, we can see that it's consistent with another study that we did in the lab, showing that there is a preferential atrophy in this region in aging. So overall, we found that cortical morphometry and hippocampal microstructure was the best predictor of aging and Alzheimer's disease progression. And for future work, we want to replicate our analysis. So we are going to use a PreventAD dataset. We are going to first look at if we can replicate our components. We want to see if we have similar relationship to demographics and cognition. We will also have access to additional scans, so the MPM protocol and the QSM. And also we will be able to relate our components to markers of the pathology per se, so CSF, amyloid, and tau. So with that, I would like to thank you for your attention. I would like to thank the institution and also the funding agencies, and of course my lab with Mallar Chakravarty, my great PI. Thank you.
Thanks a lot, Aurélie. There's one question from Dr. Martina Callaghan. She says the QC excluded quite a lot of participants. Is it known what the typical reason for a QC failure was? And did exclusion dominate in any particular group, for example, AD, MCI, or health controls?
So I don't think I tested like statistical differences between groups, but what I can say that we lost participants at different steps of our processing. It's not specific to one scan in particular, but we needed to have good T1 weighted scan, we needed to have good T1 map and good T2 star maps, of course, but also after the pre-processing, we did another round of QC to see if the pre-processing steps were working properly, as well as after the segmentation and the cortical extraction. So all these steps require different steps of QCing. So I don't have the specific number in mind, but we lost a few at each of these steps. It was not like specifically related to one type of scan or one specific processing step.
Yeah, thank you. There's also one question by Mustafa Bourara, who asks, Any reasons why focusing on gray matter but not including white matter, giving the mounting evidence of the implications of white matter integrity in dementias?
Yeah, it's a very good question. Actually, I also investigated that a bit in my stats. The main reason why I didn't include it, like I went back from using the white matter, was that I was a bit worried about the impact of the fiber orientation on my T2 star. So I decided to go back and stay in the gray matter for this reason, actually. But like with, I think like Martin Callaghan just created a new paper trying to find solution for this kind of issues. So I think like that will definitely be something I would like to do in the future.
Okay, thank you. So I think, wait, there's another question if we have time. So it's Christina who asks, I was wondering, how did you control for the age differences between your group? Did all the participants come from the same center? And if not, how did you control for the different scanner effects?
So our participants are from two distinct datasets, but all scanned with the same scanner and the same protocol. So it's from the AGB dataset and the Prevent AD dataset. But as I mentioned, the protocol is similar. So that was not a confirm that we needed to really take into account. Regarding the age, I didn't control for it, but actually, maybe I can show something. So the fact that we saw, I'm sorry, oops. Sorry. Oh, I don't have the slide that I wanted. I'm sorry. But basically what we saw in our PLS analysis was that the familial group was in the opposite direction than the NCI and AD. So it was better to be in the familial high-risk group. But I think this was purely driven by the fact that they, as I mentioned in the demographics, they were like 10 years old, 10 years younger in average compared to all the other participants. But otherwise, like we didn't control specifically for age since we included age in our PLS analysis.
Okay, thank you, Aurélie.